Now, in your previous session, we have already studied about what exactly is an alpha amino acid, how it is represented as, what are the different types of alpha amino acids, and what is a zwitterion. It exists in the form of a dipolar species having a positive and negative charge. Now, let's discuss the essential and non-essential alpha amino acids. Now, there are almost 30 alpha amino acids which are obtained by hydrolysis of protein. Now, how exactly that we are going to study once we are done with the general essential and non-essential alpha amino acids, when we will be discussing about the peptide bonds. Now, there are 30 alpha amino acids, out of which, these alpha amino acids are obtained by hydrolysis of proteins. Now, what exactly are essential and non-essential alpha amino acids? Now, these amino acids, some are synthesized inside body. Okay, some are synthesized inside the body, whereas there are some alpha amino acids which are not synthesized. I would say some not synthesized inside body. Okay. Now, the ones which are synthesized inside the body, they are called as indispensable or non-essential alpha amino acids. Whereas the ones which are not synthesized inside the body, now in this case it has to be externally supplemented by diet. Okay, such alpha amino acids you generally call it as essential alpha amino acids. Okay, now let's make a list of the essential alpha amino acids first, and then the non-essential. Then let's move our discussion with the another aspect of alpha amino acid. So the essential alpha amino acids, generally you have lysine, then you have phenyl alanine, then you have leucine, and you have lysine, phenyl alanine, leucine, and then you have isoleucine. Then we are talking about essential alpha amino acids. Among these four, which is there already. Now the next is methionine, very important. Then you have threonine. Okay, these are the couple of essential alpha amino acids. Then you also have tryptophan, and there are many more. The list is pretty much huge. So the commonly essential alpha amino acids, they are lysine, phenyl alanine, then you have leucine, then you have isoleucine, then you have methionine, then you have threonine, then you also have valine. Okay, these are the essential alpha amino acids, out of which we will be mostly we are expected to know what exactly is a phenyl alanine and what is the representation for methionine. Okay, now let's move on to the list of non-essential alpha amino acids. So the most important thing is they are synthesized inside the body. So now these alpha amino acids, they are synthesized inside the body. So let's have a group, or let's have a list of these alpha amino acids. You have alanine. Okay, and then you have many more. You even have aspartic acid. Then you have something what you call it as glutamic acid. Okay, then maybe there are many more, but we should just condense our list to the ones which are important. Then you have hydroxy proline. Okay, then you have proline itself. So you have cysteine, then you have glutamine, and then you have lysine, or you could say it's glycine basically. So these are the non-essential alpha amino acids, and there may be many more, out of which if you might have already studied about glycine, which is NH2, then you have CH2, you have COOH. This is the only alpha amino acid which is optically inactive. Okay, now as I have already been discussing that it contains a chiral center. All alpha amino acids, they contain chiral center, right? So I would simply say that these molecules are generally chiral except glycine, okay, which is nothing but NH2, CH2, you have COOH, right? So now let's try and represent an alpha amino acid. So we have this chiral center, then this is the hydrogen, and then you have the carboxylic acid group, and then you have the R group. R group can be anything, and it can be like methyl group, it can be like phenyl group and all, and then you have like NH2. So now, as per the convention, this is the chiral center, and the negative group is towards the left. So you call it as L form, whereas when you have something like this, wherein the negative group is towards the right, you call it as the D form. Okay, so you could say that this is L form and this is D form. This is L form basically, and this is the D form. Okay, so this was about the chirality of alpha amino acids. Now you might be thinking that since it exists both in L and D form, there should be a racemic mixture, which is there should be a racemic mixture. Now this point, it could be something like it could be an obvious thing that we may feel that there might be some alpha amino acids which exist in racemic mixture, but no. Since these are naturally occurring compounds, few are synthesized outside and externally supplemented inside the body, these are never obtained as racemic mixture. This has to be understood. Now this was about the essential and non-essential alpha amino acids. Now in the beginning, I have already studied that these are hydrolysis products of protein. Now what exactly do you mean by that? Let's try and understand that. Now what exactly is an alpha amino acid? We already know, right? You have at the alpha carbon, you have an amino acid, that is amino linkage. Now let's see what exactly is going to happen to your alpha amino acid. Okay, now let's say if you have something like let me orient this like NH2, and then you have CH, here you have R, here you have COOH. This is I'll just put this as R1. Okay, now this will condense with one more molecule of another alpha amino acid, which could be similar. That means it could be with the same alpha amino acid, or it could be something like different. For my convenience, I'm just taking it different, or you could even take something which is similar. So now we have a choice, right? So I will say that let me take something which is different. Okay, so now I'll write this in the same manner, but instead of R1, I'll write R2. So this is going to be NH. I'm just going to write this as NHH, and then this is CH, R1. So instead of R2, instead of R1, I'm writing R2. Then you have COOH, right? So now what exactly happens? The condensation process occurs as a result of which OH and H is eliminated in the form of water. Okay, and then you could even say that this NH there could be one more molecule this side, there could be one more molecule this side. So the chain keeps on growing again and again. So now let's represent this molecule first. You have NH2, then you have CH, you have R1, then you have C, double bond O, and then this is connected to NH, and then this is connected to CH, then you have R2, then you have COOH. So if you observe, this is nothing but a dimer, right? And this CONH bond, it is called as a peptide bond. Okay, so this is the reason you call it as a dipeptide. So if I have to define dipeptide, how can I exactly do it? When two alpha amino acids condense and lose a water molecule, that means when one peptide bond is formed, it is basically a dipeptide because two molecule condenses. Similarly, you could have something what you call it as tripeptide in which three alpha amino acids condense. Then similarly you could have a tetrapeptide, pentapeptide, and so on. So protein is basically a polypeptide. So this is the reason we consider protein to be something like a polymer molecule. So this was about the essential and non-essential alpha amino acids. We have discussed about the different essential and non-essential alpha amino acids, and we just had a look at the different types, like a group of alpha amino acids, their names. We haven't discussed their structures yet because they are not that important from your foundation point of view. But yes, it's well and good if you could just refer the structures and just have some understanding about it. Just try to remember few of them, especially phenylalanine, and then you have methionine. Then you might be coming across alanine and aspartic acid, glutamic acid, something like this. Now there is something which has to be remembered, is that all these alpha amino acids contain, so I'll write all these alpha amino acids contain amino groups, whereas the only alpha amino acids that do not contain amine group, they are hydroxyproline and proline. They basically contain imine groups. Okay, so this may appear in the exam by some or the other point of view. So you need to remember proline and hydroxyproline. These contain imine groups instead of the amine groups. So this was about the section of alpha amino acids, the essential and the non-essential one. Okay, now let's have a look at the structure of important alpha amino acids. Now all alpha amino acids except a few, like the two which I have already discussed, they contain imines, right? So this R group is going to be different in most of the cases. So glycine, so this is NH2, you have CH2, COOH. Now this is optically inactive. Then you have alanine. So alanine, you have the structure as NH2, you have CH-COOH. Now instead of this R, when it is CH3, this is basically alanine, and since it contains one amino acid, one carboxylic acid, you could simply say that this is nothing but a neutral alpha amino acid. Okay, then you have valine. Valine, it contains like you have the same NH2, and then you have CH. This CH is connected to CH, CH3, then you have CH3, that is isopropyl group, then you have COOH. Okay, then you have proline. Proline is, as I've already discussed, it is nothing but a heterocyclic compound. That means it's not an alpha amino acid, it contains an imine group instead of an amine group. So this is the structure of proline, and then hydroxyproline is also going to contain more or less the same compound, then with a hydroxy group as well. Then you have the next, which is serine or serine basically. So you have NH2, and then you have CH, COOH, and then this is connected to CH2OH, right? So if you observe, this is one carboxylic group, and you could say that the alcohols are neutral in nature, but still this is somewhat H+. So this is nothing but a form of acidic. Alpha amino acids contain weak carboxylic acid groups, so they are generally weak. Then you have arginine. So now the structure of arginine is going to be little bit different. So now let's see what the structure is. NH2, you have CH, then you have COOH. Okay, now this is connected to CH2, then you have CH2. Now in this case, what exactly happens is you have CH2 twice, and then as you could see here, you have an amino skeleton. Similarly, you also have NH, you have C double bond NH. So this is, even though it doesn't contain a double bond, but still you could say that it's not NH2, right? So this is connected to NH2. Okay, so this is nothing but arginine. Okay, so these are the structures of important alpha amino acids. You could try to remember few of them. And alanine is methyl. Then if you have hydrogen substituted by phenyl, then it becomes phenylalanine. So these are the important alpha amino acids. Okay, so now let's since we have finished alpha amino acids, the next part of our discussion is going to be proteins.